Hi, and welcome back to Sifting Through the Cytoplasm. My name is Gil Pye, and today we are going to be talking about the amazing process of genetic transcription and translation.
If I were to go out on a limb real quick and assume that you, watching this, are a human being, then I can tell you that right now you're doing something so amazing and complicated, it took lots of super smart scientists hundreds of years to figure it out.
The process that she is talking about is called transcription. It's basically when the information in your DNA gets transferred into RNA so that it can produce proteins and make you look like me. Here's my colleague Gregory, an expert on transcription, and today she's going to talk to us about how this process works.
The process starts out in the nucleus of a cell with a strand of DNA, like this one, except way, way smaller. Just for background, the DNA is actually two strands, wound together in what is known as a double helix, which looks like a windy staircase. Each strand is made up of nucleotides, which are made of three basic parts: deoxyribose sugars, which serve as the backbone, phosphates, which connect sugars, and nitrogenous bases. The bases are thymine, adenine, cytosine, and guanine, unless we're talking about RNA, which uses uracil. So anyway, the DNA is all nice and twisted together, and then along comes this enzyme called helicase, which unwinds the DNA and unzips the nitrogenous bases, basically breaking down the hydrogen bonds. Then, on the template side of the unzipped DNA, loose RNA nucleotides match up with the corresponding DNA nitrogenous bases. Uracil and adenine are paired together, while cytosine and guanine are paired. Since both the RNA nucleotides and the other side of the DNA both complement the same template side of the DNA, they should be identical, except for one difference. The DNA uses thymine, and the RNA uses uracil. Next, an enzyme called RNA polymerase joins the loose, but correctly ordered RNA nucleotides together in a coded strand of pre-mRNA. Now, before the pre-mRNA is ready to leave the nucleus, it needs to be turned into real mRNA, also known as mature mRNA. This occurs through a process known as post-transcript manipulation of RNA, aka RNA editing, involving getting rid of typos as well as adding a sense of armor. The unnecessary typos in the RNA are called introns, which are removed by the ends needed, fusing them together along with cutting out the typo. The RNA also requires armor before it can exit the nucleus, so it's given a helmet, aka a 5' cap, which is a special type of guanine. It also has a nice cape of adenines, which act as a buffer if the mRNA is hit by speeding glucose or something else in the cytoplasm. This is called a poly-A tail. Once the RNA is perfected and armored, it's ready to enter the deep, dark realm of the cytoplasm.
Okay, so now that we have the mRNA, we need to use a process called translation to build a protein with that information. In order to understand this process, I've called upon my good friend, Dr. Cytoplasm, a talented genetic linguist who's an expert in the field of translation.
Thank you, Gil. The process of translation starts out just outside the nucleus, in a nearby ribosome. Once the mRNA has exited the nucleus, it finds a ribosome and starts feeding into it like the world's tiniest dollar bill in a vending machine. The 5' end goes in first, and the 3' end is last. The mRNA feeds through the ribosome until it finds the start codon, AUG. Then little transfer RNAs, or tRNAs for short, start coming in from the outside.
Really quick, here's a breakdown of gRNA. It looks a little like this, and it has three nitrogenous bases, in this case called anticodons, which are attached to the bottom side. These anticodons determine what amino acid is attached to the top of the mRNA. There are about 21 possible amino acids.
When the tRNA enters the ribosome, it finds a space where its anticodons match the specific triplet codon of the RNA. Then it deposits its amino acid and leaves the mRNA subunit. The other tRNA continue to move through the mRNA like an assembly line. The amino acids left by the tRNA are linked together by peptide bonds and form a chain. As the ribosome moves along the mRNA, it will eventually hit a stop codon, which will then release the amino acid chain.
Boom, a protein is born.
I'm Dr. Cytoplasm.
I'm Gregory.
And I'm Gil Pye. Thanks for watching Sifting Through the Cytoplasm.
And be sure to tune in next week when we talk about the functions of the human liver.